Bismillahirrahmanirrahim. Today the topic of our discussion is chemiosmotic hypothesis. As we have seen in the previous lecture that there are 4 complexes of electron transport chain and the fifth one is the ATP synthase. Basically this ATP synthase is responsible for the transfer of hydrogen from the hydrogen ions from outside the membrane to the inside the mitochondrial matrix, and this process is known as chemiosmotic hypothesis. ATP synthase is the smallest molecular model, and this is the smallest molecular motor and it acts as a turbine. There is a development of the proton gradient outside the membrane, due to which the protons travel from the ATP synthase and enter inside the mitochondrial matrix. ATP synthase allows the movement of these protons. The hydrogen ions pass from FO subunit of ATP synthase, and the FO subunit is further composed of 10 subunits, and FO with the FO, the O stands for its inhibition from the oligomycin. This is the structure of ATP synthase. Only the writings are inverted, but the structure is totally accurate. As you can see in the green color represents the FO subunit and the rest of the structure represents the F1 subunit of the ATP synthase. So basically, if ATP synthase has two main structures, that is the two main subunits, one is the FO subunit and the other one is the F1 subunit. FO subunit, you can see in the diagram there are 10 further carbon subunits. It is divided into the 10 subunit, and with this there is a gamma handle attached, and with the gamma handle alpha and beta subunits are attached. This gamma handle with alpha and beta and omega subunits are also attached with it. These are all known as F1 subunit of the ATP synthase, and the 10 carbon subunit is known as FO subunit. This is the structure of ATP synthase. Next, you can see in the diagram that FO subunit has a lot of hydrogen ions outside the membrane, and it allows the entry of hydrogen. And how it allows the entry of hydrogen ions? When these protons pass from the FO subunit is rotated. Due to the rotation of FO subunit, there is the rotation of gamma handle, and due to the rotation of gamma handle, there is the conformational change in the beta subunits attached with the gamma handle. This mechanism is known as binding change mechanism, in which when the gamma handle is rotated, there is a conformational change in the beta subunit. So ATP synthase is the smallest molecular motor that transfers the hydrogen ions from outside the membrane to inside the membrane. This beta chain occurs in 3 conformation. Actually, the beta chain that is undergoing the conformational changes has 3 conformation. One is the open, second one is the loose, and the third is the tight conformation. Open chain is responsible for the release of ATP, and the tight conformation, that is the third one, that is also responsible for the release of ATP, and the second one, that is the loose conformation, is responsible for the binding of ADP and inorganic phosphate. So overall it is involved in the production of ATP, ADP and inorganic phosphate. Obviously we know that the ADP is further used, and it is used for the production of ATP as well. So this is known as the binding chain mechanism. Peter Michael was the first scientist to discover this process, that is the chemiosmotic process. And what is chemiosmotic hypothesis or chemiosmotic process? That in chemiosmotic hypothesis, hydrogen ions outside the mitochondrial membrane transfer to the inside the mitochondrial membrane with the help of ATP synthase, and ATP synthase consists of 3 subunits that are basically it has 2 subunits, and we can divide easily in can explain it in 3 parts, that is the FO subunit and F1 subunit. And when FO subunit rotates, then gamma handle rotates, and due to the rotation of gamma handle, beta subunits undergoes conformational change, and due to these the protons are transferred from outside the membrane to inside the membrane. So this transfer of hydrogen ions from outside the membrane to inside the membrane is known as chemiosmotic hypothesis. So this was all about the chemiosmotic hypothesis. Thank you.